Okay, so I mean, based on a lot of discussion that we had yesterday, and in the morning, I thought what I could do was allude to morphogen gradients, but then link the interaction of morphogen gradients to shape change. Okay, and some of that has been done very nicely by the work of Eric Wishaus and Adam Martin, who was in his lab many years back. And so I'll actually go through that work to first illustrate what's happening. But just to continue from yesterday, what I showed you was that in the early Drosophila embryo, there is a morphological shape change which is taking place where one sees the apical membrane as rich in villi-like structures. It forms polygonal structures. These polygonal structures are organized in various kinds of polygons with a hexagon dominance, and this changes as the development is proceeding. So what going to try to look at today is essentially after the cycle 14, where complete cells have formed, and then the first transition from a single layered epithelium occurs to form 3 germ layers. So we'll look at the transition which occurs to form 1 germ layer, essentially 2 germ layers at least, or 2 epithelial cell layers, and that is essentially formation of the mesoderm. But before we go, a lot of the work which I presented yesterday, I didn't acknowledge the people. There are a lot of people who've contributed to this, and we've also got a better understanding of how molecules flow through the system, because these are sort of open pseudo cells with modeling approaches which we have used with Amitava and Mithun. We've also learned to analyze our fluorescence data of how these molecules spread through this kind of system thanks to our collaborations with them. So when we are essentially thinking of these shape changes in the context of embryogenesis, we were talking a lot about the forces which are involved. These forces are forces which are combinatorially expressed by adhesion, tension, and viscoelastic properties of the cells, which sort of give rise to the cell shape change. But really, these changes occur often in response to the change in expression pattern of genes and proteins, which dictate not only sometimes the patterning cues of the system, but also the body axis, the anteroposterior axis, the dorsoventral axis, and so on. So what is this link? And, how can we begin to study the link between changes in protein expression and activity to that of cell shape? And I'll, of course, keep this as a talk mostly from literature and from the work, like I said, of Adam Martin and Eric Wishaus, and towards the end show you some more cellular analysis which we are attempting to do in the lab. So we saw that, for example, there could be changes in the amount of adhesion. There could be changes in the amount of tension in this layer. And these together are the ones which are actually giving rise to the shape change which takes place. Now, all of this is not happening alone, right? All of this is happening in the context of gradients, and very nicely alluded to already in the beginning by Marson. And so in the case of the blastoderm embryo, there are gradients in the anteroposterior axis, in the dorsoventral axis. These morphogens give rise to activation of transcription factors. The transcription factors often are expressed in very precise domains along this axis, and they're able to pattern those domains in that axis, right? So we want to understand how these gradient molecules are actually interacting with the cell shape change. And this is again in continuation with that. When we think of a morphogen gradient, we have, let's say, the morphogen being synthesized, but its promoter is often very complicated. Its promoter is getting feedback sometimes by not only itself, but also a combination of regulation by other transcription factors, right? And that then shapes these flags, which we are seeing in this system. Okay, so coming back to the shape change, and today we'll look at more so the shape change which occurs in gastrulation. But one could imagine shape change of these epithelial-like layers in any process. And another one which hopefully Maitri will come and talk about next week is that in dorsal closure. So one of the things which happens in gastrulation is that we start with this epithelial cell. The cross-section is polygonal, and then there is a constriction event which takes place in the apical membrane, which makes the apical membrane area smaller. Okay, so now you can imagine that we spoke a lot about constriction, contractility, adhesion changes. One of the things which may happen here is that there is a change in the actin networks which are present, and then with the help of myosin, these networks are coming together and getting constricted, right? So now, of course, we are already showing this as a schematic, but this was really found in a very seminal study in Wishaus's lab when they started addressing what are the shape changes which occur in gastrulation. Another shape change which occurs during dorsal closure is that of closing off of this membrane on either side to give rise to a complete ectoderm and epidermis, and the cells which are there in the middle here are often constricted, and they also pinch off and they give rise to this zippering off process. So this zippering off process is very similar to even what may happen in wound healing, and it's also a very interesting model where one could learn about how this zippering process occurs to heal the epithelia, and also how might the mechanics reflect what is happening, let's say, in a wound when there is an actual disruption of an epithelial cell sheet. So something which she will talk about as well. So coming back to the early embryo. Now this is exactly the time points which I was showing you yesterday. There are a set of morphogens which are added to the embryo by activities during egg development, during oogenesis. And okay, so coming back to the early stages of the embryo, this is when the nuclei are internal. Okay, they are dividing inside. But already, even if the nuclei are internal, there are mRNAs which are partitioned exactly, accurately to pattern the body axis, right? So here is the mRNA of a protein called bicoid, and here is the mRNA of a protein called nanos. These are anchored in specific locations because to begin with, they were deposited in that very location when the egg was being made inside the mother. And then there are proteins which, as a result of signal activation, they get partitioned to specific regions of the embryo. So, for example, the dorsal signaling cascade gets activated on the ventral side. This gets activated because there's a ligand which is present in the perivitelin space. This ligand in the perivitelin space binds the receptor called the toll receptor and activates the signaling cascade such that dorsal can now enter inside the nucleus. And also another system which patterns the termini of the embryo, which is called the activated MAP kinase system, which in response to, again, receptors which are activated just at the poles, is leading to a specific activation of MAP kinase only at the anterior and at the posterior. So, as much as these look like separate entities, at all the boundaries, they are likely to interact with each other. And their interactions are likely to pattern those boundaries specifically in a certain way. Now, this is an example of how these gradients would look, right? So, in the previous picture, either you look at the mRNA or you look at the protein, it's a static image. You are just looking at the concentration at that particular time point from one embryo which was analyzed. This is a live movie, two movies. One is where bicoid is tagged with a modified green fluorescent protein, GFP, called Venus. And another one is where dorsal is tagged with GFP. So here you can see this is already playing. And in this case, very soon, you'll start looking at nuclei which are lighting up in the anterior region of the embryo. So there you go. You see the lighting up and then it disappears. You see the lighting up, it disappears. And this is essentially the different cortical division cycles which we were talking about yesterday. So when you see these nice spheres which are lit, this is when the gradient is present inside the nucleus and there as well. And when you see the signal disperse away, this is when the gradient is not present inside the nucleus because the nucleus itself has broken down and all the contents of the nucleus are now present in the cytoplasm. Yeah.
So, now what is happening on screen? So it looks like bicoid concentrations are coming in bursts. So is it that the rate of transcription sort of varies as the cell cycle is progressing?
So the rate of transcription also varies, but this is largely the protein localization activity. Okay. So when you see the dots coming up, the dots indicate also the presence of nuclei. And remember that these stages, the nuclei are dividing. So when the nuclei divide, there is a point at which the nuclear envelope breaks down. And whatever components are inside the nucleus are going to spread away into the nucleoplasm and then the cytoplasm. And
the whole cell membrane hasn't formed, right? These are still divisions in the...
So these are cortical divisions. And so this is a point at which the nuclei have migrated to the cortex and you are actually seeing the nuclei. So you see also the concentration of nuclei is increasing. All these spheres which are coming up are actually individual nuclei. And when you see that the concentration is increasing, that is because it is dividing once and then it is dividing again and dividing a third time. So this is the starting point of the movie, no signal. And then you start seeing the 10th cycle and then the 11th cycle and then the 12th cycle and the 13th cycle. And you start seeing multiplication of the dots, if you will, and each of those are essentially nuclei. Okay? So the nuclei are already present at the periphery at this point. And really you can monitor the signal and how it is increasing as the divisions are taking place. So it turns out that as the divisions take place, even the amount of signal which is present does increase over time. And this kind of method allows you to quantify the amount of signal across the division cycles in this syncytial embryo. So again, starting here, this is the first division where the nuclei are present, then 11, then 12, then 13, and you have this, and especially, after that, the nuclei elongate in cellulization, so you're seeing that as well. But you are seeing accumulation of the signal inside the nucleus. That accumulation of the signal inside the nucleus happens in the interphase stage of the cell cycle. All right? So, okay, so this is going on and this happens before gastrulation takes place.
So, one thing to clear, this is the mRNA firstly.
This is the protein, this is the protein. Both are proteins.
So, mRNA is inside the nucleus.
So, mRNA is made inside the nucleus and then it comes out. And then that is also something which one sees increasing over time. And but this activity is being read out of the fluorescently tagged protein.
So it seems that when they're dividing, that is the point that the gradient seems to break down, or it is not there.
Well, it is still there. Okay, so that has also been measured. It's just shallow.
It's just...
People have measured the gradient, but that is shallow.
And what is venous?
Venous is a modified version of the GFP tag. Okay, so it gets excited. So GFP itself, in the way we use it today, gets excited by 488 nanometers. Venous gets excited by 514 nanometers.
Is it because of the diffusion that is happening after the formation of protein, because the signal is going away?
Yeah, so is it diffusion? Is there a gradient in metaphase when there are no nuclei? This has been analyzed quite carefully, And what is known is that the gradient is still there, it is just shallow. It's a very sharp gradient when it is there in interphase, and in metaphase the gradient is
Gradient
of the protein. So this one is also, so why is this only in one part of the embryo, right? This is not there in this part. So because it is the protein which patterns the anterior part of the embryo, and because it is concentrated in this region, it is leading to gene expression changes to give rise to the anterior region of the embryo only here, not here. And this is the one which is cycling. So there is a gradient, right? So there's high concentration here, low concentration here.
So my question was regarding the disappearance of the signal that is happening.
So even when it disappears, there is still a gradient. From anterior.
Why does the fluorescence does not basically?
So the fluorescence is because you have adjusted the scale to read out the maximum intensity here, and which is the maximum intensity inside the nuclei. So your eye is not tuned to looking at the fainter signal. And also the color scale which is being used here is not tuned to looking at the fainter signal. But there is a gradient. It's a shallow gradient, but it's there. Okay?
Just one more question. So you did mention that this bicoid protein which is being produced is produced by the embryo, right? Is it?
The bicoid protein is produced by the embryo by and large. There's a very small amount of protein which is present in the embryo as well.
Yeah.
But it is produced when the when the development starts. Yes.
So because there is maternal, mRNA.
So in the case of bicoid, most of the carryover from the mother is the mRNA.
Yeah.
In the case of dorsal, most of the carryover or what the mother gives is the protein. Okay? So there are different forms of how the maternal effect occurs. The maternal load in this case is the mRNA, and the mRNA starts transcribing to give the protein in this axis. The mRNA is also localized more carefully only at the anterior.
So this venous tag, is it also present on the maternal mRNA? So in the sense
Yes,
that,
of course.
so okay.
It's also present on the maternal. Okay? Okay, so now we have these two kinds of gradients which set up. And one of the things which we want to understand is, at what point do we clearly see an interface between the gradients and this kind of shape organization or change in this kind of shape organization? So just a reminder from yesterday, where embryos are single cells and they divide and they give rise to an organization of an epithelial layer which is polygonal in nature and multicellular as an array. And if we look at different kinds of embryos, at some point, all of the different kinds of embryos that we've been thinking about and learning from, the mouse embryo, the sea urchin embryo, the Drosophila embryo, C. elegans embryo, they all turn into this sort of epithelial-like layer organization. And so we are moving from a point where there was a single cell to a point where there are multiple cells, and these multiple cells are just present in one layer across the embryo. All right? And what we want to understand is, what are the shape changes which occur when the layer becomes from 1 to 2 layers. All right? So morphogenesis, change in form in the early embryo, and what we are going to look at are change in shape which occur during gastrulation. And Louis Wolpert said this very nicely because he studied this process a lot. It's not birth, marriage or death, but gastrulation which is truly the most important thing in your life, because that is the first time you become a double layered embryo. Okay, so when we look at the formation of germ layers, there are some interesting differences between vertebrates and invertebrates. All right, so even though they are both, broadly classified into 3 kinds of germ layers, what we see here, which is a commonality, is that the ectoderm, which forms the skin and the nervous system, is present towards the outside. The nervous system is what is different. In vertebrates, the nervous system is here on the top, on the dorsal side, and in insects, the nervous system is on the ventral side. And the endoderm, which is present internally, is responsible for giving rise to the gut, liver, lungs. The mesoderm, which is next to the ectoderm, is responsible for giving rise to broadly the muscle and heart and so on, right? So such tissues. So, once these layers form, so, this picture almost says that these layers form and they are now different from each other, right? So how did we know that these layers actually maintain as different from each other? And how is there no mixing between these layers, especially when tissues have not formed? So this is around the nerve cord, and essentially what we are looking at is the skeleton and the muscle.
Yeah, but you got a paler shade of red also.
Yeah, so I don't exactly know the key here, right?
Okay.
What has been represented in these different colors. But by and large, all of these colors are represented by this, okay, including the blood, okay? Okay, so once the germ layers are formed, there is no tissue formation yet at that time. But these germ layers are distinct from each other. They do not mix. And so then what is the property which actually keeps them apart? And some of these experiments were done a long time back. These experiments try to understand what are the properties of the germ layer cells which keep them apart, right? So suppose I had an artificial scenario where I take cells from the ectoderm and the endoderm and I mix them up or keep them side by side. It's not like they will mix with each other. They remain separate, okay? And you will see that they will separate out really and in a manner that all the endoderm cells will be together and the ectoderm cells will be together. Another way of doing this experiment is actually by disaggregating the cells, right? So now, do the cells have a memory of their location, and will they find their partners that they bind with, right? So one could take, for example, let's say ectoderm cells from an amphibian embryo, and one could take mesoderm cells from an amphibian embryo, dissociate them. So now dissociating means that the cells are not dead. They are very much able to perform their function, but they are not bound to each other anymore. And then mix them, right? So now the question is, what will be the way in which they separate out, right? And what is seen is that they will separate out such that the ectoderm, which is blue, is inside, and the mesoderm, which is gray, is outside. And this almost always happens, all right? The mesoderm will be outside and the ectoderm will be inside. And obviously there's some physical property here which is allowing them to recognize their own cell type, and also another physical property which is allowing them to go inside and then be covered by the mesoderm cells. This is not the actual situation of the embryo, right? In the actual situation of the embryo, the mesoderm is inside, the ectoderm is outside, right? So there may be some other interactions which are also responsible for this, which have been discovered lately, but they are also dictating this kind of phase separation. But there is a very interesting phase separation which is taking place, right? And the question could be, what does this phase separation depend upon? And a long time ago, one of the ideas which was tested was to understand if just difference in adhesive properties could cause this kind of phase separation. So one of the ways could be difference in adhesive properties, all right? So what is lately found though, even before discussing this experiment, I would like to allude to that. What is lately found though is that there is a both difference in adhesion properties as well as a difference in contractile properties of these germ layers, which actually causes them to be apart from each other. All right, so Steinberg tested this. What he did was he basically expressed different amounts of cadherin, in this case N-cadherin, in cells, in the same cell, but different levels of cadherin. And what he found was that in those cells where cadherin was higher, the cells ended up being inside. In those cells where cadherin was less, the cells ended up being outside. All right, so just by changing the amount of adhesion, one could separate cells into two different populations, right? Highly adhesive cells are inside, less adhesive cells are outside. And so then, is this the way in which this kind of separation takes place, that when you disaggregate the cells, that the ectoderm cells go inside and the mesoderm cells go outside? It is a likelihood, one of the possibilities. It's been found recently that in the case of zebrafish embryos, it's not the adhesion but actually the contractility of the cells, and along with that, the property of these ectodermal cells to interact with the outside environment, which actually puts them in the position that they are in, right? So the real embryo, the ectoderm is outside. It may have increased tension and therefore it is going inside as compared to the mesoderm, but it also has this property of interacting with the environment, right? Sometimes the white line membrane or the perivitelin space. So there are many other factors which regulate in the way the real embryo cells are organized. But in gastrulation, of course, there is a point at which the mesoderm cells are going to be present inside the ectoderm cells, right? And so in all of this, again, we are talking about the same sets of molecules which we spoke about yesterday. We are talking about the contractile molecules. So I'm showing them with a different picture today, where we are looking at actin and we are looking at myosin motors. Myosin motors, you can see, bundle these actin filaments, and they're going to place these actin filaments in a certain location inside the cell if their activity increases. And we are looking at the integration of this entire system with surface molecules, and here we are talking about the adherence junction molecules like ecadherin. A lot of times, ecadherin can be present on its own. It can be present bound with neighboring ecadherins, and this extracellular domain is what actually brings the neighboring membranes together. So ecadherins also have this property where they will activate the actin cytoskeleton because of interactions with a whole host of molecules which are called as catenins, which organize the actin cytoskeleton. And the actin cytoskeleton itself can also have differing organizations based on the activation of myosin. All right. In addition to this, there are other actin regulators which regulate how much bundled or branched actin is present in the periphery of the cell. So in a sense, we are now adding more molecules to this system which regulates the cell shape, but by and large, essentially just looking at adhesion and contractility as forces which change when the shape change is different in different stages. So what's happening in gastrulation and specifically in the Drosophila embryo is that we are at a point where we have this kind of layer of cells, and then we are at a point at which some region of the cells constrict in one location and then they're moving inwards. Okay? So this constriction, this apical constriction occurs so that the cell shape change can take place. All right? How do we begin to understand what are the changes in adhesion and contractility which occur at this point to allow for this very spatially controlled shape change in this kind of system? So not too long back, this entire exercise of what is the volume of these cells been estimated exactly in this system. And it has been found that the volume is conserved.
So one looks at Z-stacks and try to...
Yes, you can do Z-stacks. You can basically do a 3D projection, estimate the volume, and try and figure out the volume in exactly all of these different points, and the volume is conserved.
So that is why the basal side sort of...
expands. Absolutely. Yes. So this one you're seeing as expanding, this one is contracting. It also becomes a little longer, the entire cell.
And then if the two sides sort of pinch together, that will form a tube along the length of where the contraction.
If the two sides pinch together, what does that mean?
Like the sides flank.
Oh, yes, absolutely. So if this was the case that, I had other forces, it could give rise to a tube. Yes. Okay, so Drosophila gastrulation. So what are we looking at? We are looking at this movie, right? This movie occurs, so these were the first divisions that we were talking about yesterday. After these divisions, a complete epithelial cell has been formed. This epithelial cell is long and tall, and it is about 35 microns tall. And after the formation of the cell, there is a particular region in the embryo, which is that of the ventral furrow, which is what we'll talk about more, where constriction takes place and gives rise to invagination of these cells. There's also this other furrow which forms at the same time, which is called the cephalic furrow, which is essentially happening ahead of the head region. Okay, so we are basically talking about this region. And if we look at, there are very many different ways of looking at this process, right? So the same elliptical embryo, one could cut in the middle and make sections and look at the dorsoventral axis, right? So this is now the dorsoventral axis. This is dorsal, this is ventral. Why do I know that? I know that because this furrow, which gives rise to the mesoderm, is present on the ventral side. This is the place where dorsal is present, and what you see here is essentially transcription factor twist, which is present only in these cells which are changing the shape. Okay, and then finally you see that the cells which have gone inside are the cells, of course, which also still express twist. What is the kind of shape change that is occurring? You have a uniform layer, you have apically constricted layer, you have these cells becoming elongated, and then they're going to pinch off and essentially go inside, right? They essentially settle on top of the cells which form the ectoderm. Sorry?
What triggers the apical constriction?
So that's exactly what I'm going to show you, okay? That is actually what I want to talk about. So this is akin to what is called as epithelial to mesenchymal transition, where from a single epithelial layer, we have a point at which this cell does not want to contact with these neighbors anymore. It loses contacts and moves inward, right? So a process which occurs in disease and a process which normally occurs in development for giving rise to various different processes in development. What's happening in gastrulation? Right, so a very old review, but still a very nice and relevant one for this process. So what we're seeing here are different colors and different germ layers represented, right? So now these colors are represented when the embryo is just a single layer of epithelial cells. So if we look at the fate map, of the different regions of the embryo, what you can notice is that the mesoderm cells, which should be inside, of course, to begin with, are present outside. All right? And they are essentially present here. And these are the ones where twist and snail get activated. And I'll show you the signaling cascade very soon. These are transcription factors which get activated because of dorsal. Dorsal is present in this region. It leads to increase in transcription and translation of these transcription factors, twist and snail, and they are then seen in this very region, and this is the very region where the shape change occurs. Many different changes occur. One is, for example, the nuclei also are getting pushed down as this apical constriction takes place. So the nuclei get pushed down. There is a constriction of the apical membrane, like someone noticed. This one also expands. The volume is conserved. Then there is a point at which there is a change in shape, and there is a point at which these cells go and essentially settle inside. Now, at this point, the ectoderm is nicely outside and the mesoderm cells have gone in, right? That's where you can see them. They have gone in, and these cells are settled inside and they start dividing again, right? So division was halted up until now. A single epithelium is formed, these cells go inside, and only when they have gone inside and completely of a different shape, they start dividing again to populate that particular germ layer. All right? So what are the different things which we know about in the embryo at that time? What are the different genes which are regulating something like this? So one could very, very broadly divide them into maternal genes and zygotic genes. All right? So maternal, as in the ones which come from the mother, they are also the category which one could say are ubiquitous. They are present everywhere. A lot of these are present as mRNA as well as proteins from the mother into the embryo. All right, so they turn out to be many of the architectural proteins, right? So actin, myosin, also dorsal, which is coming as a protein from the mother. The zygotic genes, which get activated also when complete cell formation or cellularization is taking place, are the ones which get elicited or induced in response to dorsal. So dorsal is the one which induces the expression of twist, snail, fog, and many others, that still remain to be known, even if we are looking at about 25 years later. And these are the ones which are coincident with the shape change which occurs. So what do these transcription factors do to give rise to this shape change? Do we know what are their activities which actually give rise to the shape change? Suppose I remove these transcription factors. Am I in a position to understand what is the defect in the shape change that occurs in those mutants? Right? So that is essentially what Eric Wishaus's group did, and something which I will share with you. So to look at another way of representing the signaling pathway, we have dorsal on the top, gives rise to the activation of twist and snail. This is occurring only in the ventral domain, few cells at the ventral region, leads to activation of fog, which binds to its receptor, and then gives rise to a G protein coupled signaling cascade, which activates the G protein, activates Rho-GEF. Rho-GEF is a protein which increases more active Rho inside the cell. A small GTPase is Rho, which when it is in the GTP bound form is able to activate kinases. Here, this is the one which is called Rho kinase. Rho kinase goes and phosphorylates the small subunit of myosin, which is called squash, and that leads to increased contractile ability of the myosin, and that leads to increased contraction of the actin and therefore apical constriction. All right, so this very involved signaling pathway, of which we are showing the most famous components, there are more, that is responsible for leading to apical constriction in this region. Okay, so there are two sets of studies which were beautifully done to try and understand what is going on with this pathway which leads to apical constriction. All right, the change in shape. And I'll show you one of them. So I hope this plays. Yeah. So this one is now looking at cells exactly in the region here where the constriction is going to take place. All right, and I'll show you the movie and we can discuss what do you think is happening with these cells. It's a really nice movie. And it's only showing the cell membrane, right? When you're looking at this movie, you can imagine that there are organelles there. I mean, we look at mitochondria at this stage. And there are so many other entities inside the cell which might also be changing their distribution and function. So it's very interesting to try and understand all the dynamics which occur at this point. But when you look at just the membrane, it allows you to focus on the shape change which is occurring of just the membrane, right? And I want you, for example, to tell me, what do you notice? What do you notice which is taking place in this wonderful movie by Adder? The middle region is getting constricted. The shape change is occurring, right? And so essentially the cells are contracting and they are most likely going inside, right? Now, can we quantify this kind of shape change, right? So if we look at, for example, this kind of shape change, we can get quantitative estimates of how much the apical area is changing. And what one sees is, of course, there is a change in apical area. That is what is expected when the cells are constricting in the apical region. Now, if we look at one at a time, if we look at many cells, you can't see the activity which really happens. But if we look at one constriction event at a time, one cell at a time, then what one sees is that it is constricting in batches. Okay, so there is a constriction pulse, then there is a stabilization pulse, S, there is a constriction pulse, there is a stabilization pulse, and there is a constriction pulse, right? So there are phases of constriction and stabilization which are alternating with each other to finally give rise to an apical constriction event in each cell. Now, while that is happening, if I look at the constriction rate of these cells, they are not just constricting smoothly, okay? There is a point at which it constricts and relaxes. There is a constriction phase, then there is a relaxation in the middle. There is a constriction phase, and then there is a point at which it does not constrict, and so on. So this constriction is essentially coming in pulses, all right? And the pulses are important because the pulse in which there is high constriction is also the time at which the apical area reduces quite a bit. When the constriction is no longer there, so sorry for the use of the word relaxation, that's not correct. When the constriction pulse is no longer there, then at that point there is a stabilization of the area. So it is decreasing, but there is a phase in which these constriction pulses are coming about. Okay, and really, how does this kind of pulse correlate with the activities of the contractile molecule is what Adam did next. So he noted that when we look at all of the different cells, so let's just, for example, point to this cell. You see it's in sort of the yellow-orange region, which means that it has a high constriction rate. Then it moves to a point at which there's a low constriction rate, and then again it moves to a point where it has a high constriction rate. Right? So, and that is the case with all of the cells. They are not all constricting together. Okay? They are constricting in their own cycle of constriction and no constriction. Right? And one can do that by following the constriction rate of all of these cells. So that is one point. The other one is, how does myosin look in this point when the constriction is taking place? So it turns out that one can essentially just compare a point at which the areas are more and a point at which the areas are less. And what one sees is that myosin is present as a coalescent accumulation in the center. All right, so there is an increase in myosin in the center of these cells. And what one sees is that as the constriction proceeds, the apical area is reducing, myosin intensity is increasing. So there is a correlation between the amount of myosin as these two activities take place. There is also a correlation between the increase in myosin and the decrease in area, right? So one sees a flood of myosin signal, which is essentially looked at with this tagged molecule. This spider is just looking at the membrane. This is a kinase which marks the membrane very nicely and uniformly and is used to essentially read the membrane activity. Okay, so now, what about the way in which this takes place, right? So myosin accumulates, first of all, at the center. It's not at the periphery. It is accumulating at the center, most likely linking the actin filaments in such a manner that it allows for this constriction pulse to take place. The contraction or accumulation of myosin also correlates with the point at which the constriction phase is taking place. And then there is maybe somewhat of less myosin intensity when the stabilization phase is present. So there is increased myosin here, and then there is maybe slightly less, it's still on the higher side, myosin which is present in the stable phase. What is the status of adhesion here, right? So now when constriction is taking place, if we look at myosin, there is a coalescence of myosin right here. And when we overlap this with the signal of the cadherin, which is obviously present on the membrane, what one starts seeing is indents which form on the membrane. So there's a fold or indent which is forming on the membrane, and this is coming close to a point at which the myosin is constricting, right? So it looks like there is a particular coordinate interaction of this entire layer where increased accumulation of the myosin in the middle is also correlated with, constriction of the entire membrane, where indents of myosin or cadherin can be actually seen close to the myosin accumulation. So this is essentially just very roughly said as adherens junction bending.
When the cells are constricting, so does the actin contract also increase?
The actin, so there is increased myosin and actin contractility is of course increasing. Actomyosin activity is increasing. What drives the asymmetry of myosin distribution? Very nice question. That is because myosin is getting activated only over there. And it is getting activated only over there because there is a relocation of a lot of proteins to this apical region. And one of the proteins which is actually getting relocated in the apical region is like ROGEF. And so this ROGEF molecule gets relocated, and that is coincident with the activation of myosin just over there. So increase in ROGTP just in the apical region and increase in myosin activation just right there. Okay. So exactly this signaling cascade. So like he said, what's going on? How is it just happening, in that spatial location? It's happening because of this molecule. There's an activation of this signaling cascade also in that location. ROGEF gets activated in that molecule, and because it increases local Rho, that gives rise to local activation of myosin and those pulses and leads to apical constriction. All right, this exact same mechanism. So now, what's the role of twist and snail? Right? Do we know what twist and snail do? We do know that they are there. Right? They give rise to the presence of all of these molecules. But if I remove them, then what is the consequence on this entire constriction cascade? So it turns out that if one removes twist or if one removes snail, there are two differing effects which are actually seen. So if one removes snail, then if you look at these cells, really there doesn't seem to be much of a constriction pulse. There is some, increase and decrease of area which is seen, but the apical area does not change at all. All right. When one removes twist, on the other hand, then one does see these pulses. But if you look at, there was a constriction phase and then that should have been followed by a stabilization phase. That stabilization phase is not coming up at all. All right. So twist is responsible for stabilizing the constriction, and snail is most likely important for even just having constrictions in this system. Right? And that's all that we really know at the end of the study. Yeah.
The twist knockout.
Yeah.
The twist knockout, is it trying to recover the geometry? Because the red line seems to be going up.
So basically, again, we just compare it to, the control right here. So we have a constriction phase and a stabilization phase, a constriction phase and a stabilization phase. And in the case of twist, you do have some constrictions and then again it comes back, right? You have constriction and again it comes back. So yeah, in a sense, basically the stabilization is not occurring at all. Okay. Maybe there's some small amount of decrease in area, but nearly not as much as the control. So then by the end of the study, what Adam and Bischoff's paper concludes is that these morphogens, right? So dorsal gives rise to activation of snail and twist, and they are responsible for the cell shape change as follows. So one is looking at the apical region of the cell. The apical region of the cell has all of these actin filaments. The actin filaments are getting bundled together with each other with the help of myosin coalescence. And the myosin coalescence occurs such that there are regions where there is intense myosin, which also leads to adherens junction bending in these particular regions. And there is a phase in which contraction occurs and then it stabilizes, and then again there is contraction and then there's again stability until the final decrease in area is achieved, right? So this is the mechanism by which the contraction or constriction takes place in the apical membrane to give rise to a smaller epithelium. And when we think of the transcription factors, so remember these are transcription factors. What we're looking at is really a downstream consequence of removing these in mutants. In this case, in RNAi, right? So when we remove snail, we do not see any constriction to begin with. When we remove twist, we do see these contraction-constriction phases, but we do not see the stabilization phase at all. Without stabilization, the area of the membrane does not decrease so that it can decrease again in a consistent manner. All right, so that is what these transcription factors do. Yeah.
Of actomyosin constriction. So how
So
exactly
in this
do they
case,
act?
essentially what is happening is that there are these actin filaments, okay? And what these motor proteins are doing often is bringing together a lot of the actin filaments. And since the actin filaments are, contacted not just with each other, but with also actin filaments across the cortex, this is also going to have microtubule activity. We are not even talking about that right now, okay? Because we are simplifying the story to just look at what actin might be doing. We have these actin filaments which are being bundled together with myosin, and because all of the system is a network and attached with each other and finally attached to the adherence junction, you are seeing a constriction.
So myosin is a sort of a bridge between actin filaments, right?
Yes, it is a motor protein which is sliding the actin filaments against each other.
So during the stabilizing phase, can I say that there is no myosin activity, but so apart like in the wild type, if in the stabilized phase, there is no activity of myosin, but still myosin is present between the actin filaments. Can I say
So
that?
you could, until we know more about, what is the rate at which it is getting activated and maintained in that phase, we won't be able to, comment on this. So what's going on between this and this state for myosin itself, right? And there is some change in distribution of myosin for sure. But in terms of whether it is active or inactive, is new activated myosin being added over here? These are questions which have not been sort of carefully looked at.
Okay. And in the microscopy videos you showed the green labeled myosin, the labeled myosin, it's active myosin, right? Or is it just
The
myosin?
full myosin.
Okay, even if it's
I
inactive, it
cannot
would be-
look at active myosin.
Okay.
Okay, we are not in a position. Active myosin right now, what is known as this phosphorylated myosin. And what is active myosin? Active myosin, so myosin has a light chain and a heavy chain. The light chain is the one which gets phosphorylated by Rho kinase and is the one which is forming the active myosin for now. It is getting activated in response to this Rho kinase. And can we look at only phosphorylated myosin? There are many ways people have done such things where they have, for example, looked at an immunostaining or an antibody just against phosphorylated myosin, but
You need to fix cells for that.
they do not work very, very well.
Just one last question. So, can I correlate activation of myosin with myosin coalescing? Like, the way it condensed. So, it's condensed.
So what is known is that if myosin did not get activated, you will not see this kind of accumulation.
Yeah.
That way we can correlate. Okay? Okay, so that is what is happening at the single cell level, right? But this is an array of epithelia. And then, after doing the study, Adam Martin again went and started looking at what is happening to these cells in the entire array. This is not alone, this cell. So if this is constricting, then the neighbor also must be doing something, right? So what is happening to the entire array? And this is the other paper, which came a little 1 year later, where, for example, if we look at the same movie, right? So you might remember the movie. You look at the way the shape change occurs. The shape change occurs in a very coordinated and anisotropic manner. Okay, it's not changing from all directions. If you look at this shape change, it is, if anything, it is compressing in one direction. Okay, it's not going all together like this. It's compressing in one direction. So basically it's in the anteroposterior direction where you see a greater amount of area as compared to the dorsoventral axis. The dorsoventral axis is the one which is actually constricting more than the anteroposterior axis, right? So any kind of uneven shape change can be represented by this measure, which is called anisotropy, where what they're trying to measure is just, the diameter which is in this axis as compared to this axis, and the change in this diameter in the long axis versus the short axis will give you the rate of change of this particular structure or shape of this membrane. And if you see that as the area decreases, there's also an increase in anisotropy of the cells, which means that the cells are becoming smaller in one direction as compared to the other direction. Okay. So now this is something which is happening along the entire tissue, right? One can look at one cell, but it is happening along the entire tissue. And this makes us wonder whether there is a force which is a tissue-wide force which is occurring in this particular system, right? And just by looking at this as well, we can start guessing, where is the force? Is the force in this direction, in the vertical axis, or in the horizontal axis? What do you think? Where is the force? Is it in the vertical axis or the horizontal axis? So it's stretching in the horizontal axis, so maybe the force is in the horizontal axis.
But if constriction...
And then it is regulated by the anteroposterior signaling pathway? No, actually they proved that right here. But, yeah, go ahead.
Yeah, so it should be vertical.
It should be vertical. It could be either, right? And really, this is the compression which is taking place. So they look at, these are like triple mutants of bicoid, nanos, and torso, which regulate the anteroposterior axis. When they remove all of these patterning factors of the anteroposterior axis, what they see is that there is no change to the rate at which this constriction is taking place. So the signals of the anteroposterior axis are not involved here. Okay, so definitely those signals are not involved. If I remove dorsal, for example, anyways, there's no constriction which takes place. So definitely dorsal is involved. We can't remove dorsal to learn anything much more about it than the fact that dorsal is important for inducing this kind of constriction. Okay. Now, another interesting experiment they noticed, which was happening on the side, was that when this kind of constriction takes place, and now you're looking at, just myosin as a tag. So you see these little spots of myosin which are getting intense as the constriction is taking place. And then finally, these cells have constricted and they go inside. All right. When I have a mutant here along with myosin, this is a mutant which also is defective in adhesion between cells. Okay. So now these cells are not adhering to each other very well, but the force is also likely to still be there. All right. It's a tissue-wide force. We want to understand, is the force in the vertical direction or in the horizontal direction? And here is one mutant data. This mutant data is the one which tells us that now adhesion between the cells is not there. We know that because this mutant actually abrogates adhesion. And what one starts seeing are tears. Okay? Tears which are occurring in this direction. Okay? So when you start seeing tears in this kind of direction, you then know that that must be the place in which the force is occurring. Okay? The force is most likely in this direction. Right? In the same direction in which the anteroposterior axis signals give rise to the anteroposterior axis, but they are not responsible for this force. All right, so you remove the adhesion between cells and you start seeing tears. And you can do this in a very nice way where I can just draw a line here and I can look at this line across time, and what I see is that there are cells which are intact on top, and when we see these cells across time, you start seeing tears. All right? So now, this tells you something about what is the direction of the force. It allows you to guess the direction of the force, right? Remember that we are still not looking at the plasma membrane here. You could ask, but how is the plasma membrane? Is this really a tear? Is it a hole which is being created in the embryo? Or is it just a stretched cell? All right? So it is a stretched cell. That is what it is. And hopefully I have a picture. So how do you figure out, what is the direction of the force? So again, one can do the same kind of laser ablation experiment, which I was talking to you yesterday about, where I can create an ablation zone in the anteroposterior axis, or I can create an ablation zone I can create an ablation zone in the vertical one to test whether the anteroposterior axis is the place of tension, or I can create a cut in the horizontal direction, to test whether the dorsoventral axis is where the force is. And what you see over here very clearly is that the displacement of this tissue is way more in the anteroposterior axis cut, right? So in this cut. So then this already tells you that this is the one along the length of the furrow where there is a force which is acting in this particular tissue. Yeah.
Question. So in the last slide, you were talking about a myosin mutant, right? Was it a myosin mutant or
No, this is an adhesion mutant.
adhesion mutant?
Yeah.
Okay.
Basically, you can consider it as a cadherin mutant. Okay, that's what I said. So you're reading just myosin over here. This is myosin GFP, and this is an adhesion mutant. Okay? And when you have an adhesion mutant, you start seeing tears. They could have torn in any which way. The tear is in a specific direction, which is very similar to how this experiment is done, where I have a cut which is made in this direction, and which implies that the force is in the horizontal axis, okay, or the anteroposterior axis. We don't know the origin of this force, but this is the direction of the force, right? Up until now, we do not know the origin of the force. But from the previous analysis, we do know that snail and twist play a role in at least changing how myosin looks, right? So is, for example, twist responsible for this, right? And okay, so here is how, for example, the cells look. I only showed you myosin. Myosin is looking like it has gaps over here, but the cells are completely stretched, right? So adhesion is less in these mutants. And what you see is that there is a stretching of cells in this particular direction. All right? And what I'll straight away go to is an analysis of what creates this force, right? So, for example, when I remove armadillo and twist, armadillo is the one which removes adhesion. When I remove twist, then I start seeing that in the mutant where there was no adhesion, now there are no tears. Okay? So let me go about this in just once more so that I can explain the entire complexity to you, okay? So what we began with, and we'll start from there. So just 3 things to summarize this entire story. What we began with is the way in which this constriction is taking place, right? So the way in this constriction is taking place is that it is not isotropic. There is a constriction which is taking place in this axis more than this axis, right? But we know that constriction is taking place and reducing the entire area, right? So the question is there a tissue-wide force? And if there is a tissue-wide force, then how can we identify that force? All right? 2 ways. One is, can we remove the link between adjacent cells? Can we make the link between adjacent cells weaker? So this is a mutant which removes or makes adhesion between cells weaker. It's called armadillo. And what you start seeing are tears where there is no myosin in the middle, and there is a stretching of the tissue which is now coming about, right? So when we look at the membrane, you can see that the membrane is stretched out in these regions. So this tells you that most likely the force is in the horizontal axis. You can look at really is the force in the horizontal axis by doing ablation experiments, where one is now ablating either in the vertical or the horizontal axis. If I ablate in the vertical axis, I am breaking the force in the horizontal axis. If I ablate in the horizontal axis, I am breaking the force in the vertical axis. And the amount of displacement of the tissue tells you how much tension it is in and what is the direction of the tension. So this tells you that the direction of the tension is in basically uncovered by this experiment, where I am breaking the force in this particular axis by making a vertical cut, right? So now that plus the mutant experiment tells you that it is this direction in which the force is. And what mediates this force? What mediates this force is again twist. So when I remove twist, there's no force over here, okay? And I do not see these stretched cells at all, right? So coming back to a full summary, I won't go into more details of this. You can actually look at the paper for this, for more details. A full summary of this is that one has constriction which is taking place. The constriction occurs because of increased activation of myosin in each cell. Each cell is showing increased coalescence of myosin along with actin in central regions. This gives rise to a decrease in apical area across the apical membrane. And this occurs in myosin pulses, right, in constriction pulses, where the pulses are leading to a constriction of the membrane and a stabilization of that area. And the constriction is regulated by snail, whereas the stabilization of the constriction pulses is regulated by twist. So twist is not only responsible for stabilization of the pulse, but is also responsible for allowing to transmit that entire pulse stabilization across the entire tissue. So when twist is not there, there is a forced transmission across the entire tissue, which does not take place. That forced transmission is occurring in this axis, in the horizontal axis, and is very important for all of the cells to constrict together in that particular region. All right, so this kind of study allows you to look at what are the cellular activities which take place in this scenario, and what is the rate at which these changes are occurring in response to the gradient molecules. Right, so this is essentially what I wanted to show you from literature in terms of how the gradients might regulate the way in which this kind of constriction is taking place. All right, so any questions for this? Yeah. So the mechanism itself is not clear, right? So we know that twist is a transcription factor, and we know that when twist is not there, the constrictions do not stabilize, right? And the area expands again. We know that that happens. But exactly which activity of twist gives rise to the stabilization of the cell at the molecular level is something which is not known. Okay, so I'll just for 5 minutes end with something which we are doing. So this is another way of looking at the dorsoventral axis, right? So one can take movies like this, and this is something which we are doing right now. It's not published. So I'm actually sharing data from the lab, which is ongoing. This is the dorsoventral axis. The way we do this is, and the way even the other pictures were available, is that we are able to place the embryo on its posterior side and then image a cross-section right here. So the cross-section is a circle, And when you start looking at this, this is the point at which the individual cells are formed, and you see a furrow in this specific region on the ventral side. This is exactly the place where the apical constriction was taking place. So far we've been looking at the plasma membrane here. Now in this particular study, what we are looking at is mitochondria, right? And if you look at the mitochondria, so this is looking at it in this scale, which is just telling you the intensity of mitochondria. So mitochondria are really small, like about 200 nanometers to 300 nanometers. They're really small structures present all along in each cell. And when you see this particular place where constriction takes place, there is increased accumulation of mitochondria, and these are electron microscopy images which show you that there is a huge amount of mitochondria which are actually accumulating in this region right here, right? So there is an increased accumulation of the so-called powerhouse of the cell organelle in the same place where the apical constriction is taking place. And one of the projects which we've taken up in a major way is to try and understand what is the function of mitochondria in these particular places where the constriction is taking place. So I'm only going to show you that there is a change in distribution to allude to the point that we do not know all the controls which give rise to this apical constriction on the ventral side mediated by twist and snail even now. And it's of interest to try and look at various different aspects of the cellular dynamics in gastrulation, all right? So when we look at the same movie in snapshots, what you can clearly see is that there are these red pixels which says that there is a high concentration of mitochondria over there. And what is interesting is that mitochondria seem to be accumulating at the apical region even before one can seemingly see the cell shape change occur in these regions. So there is an increased intensity of mitochondria. Mitochondria accumulate mostly in this region, which is going to give rise to the mesoderm, and not so much in these regions, which is going to give rise to or remain as the ectoderm. Yeah.
Killed by these biologists or?
Yeah, so there is not much biogenesis at this point. He's asking if the local increase is because of biogenesis. No, there isn't much biogenesis. This entire process is just taking place in 12 minutes, 15 minutes at the most. And this is translocation of mitochondria. What we also know is that mitochondria are moving from inside the cell to outside the cell. And this translocation is coincident with the formation of the ventral furrow. There is some active transport. It is microtubule driven. So they are migrating on microtubules to the apical location, exactly where the...
How is it known that it's on microtubules here?
So we've done experiments where we, for example, abrogate microtubule function, and then we see that then the mitochondria are not going there anymore. And of course, the ventral furrow itself is defective. Yes. So there are in fact two migration events. One migration event occurs during cell formation, and then there is a second migration event which occurs in response to this signaling cascade to move mitochondria to the region where the apical constriction occurs, exactly where myosin occurs. So mitochondria are also present right there and play a small role, if not, the major role in regulating the kinetics of the constriction.
So the mitochondrial translocation is happening from cell to cell?
Is happening in each cell from the basal region to the apical region. Mitochondria don't move from cell to cell. Okay. They don't. So this is also a point at which the full cells are formed. Mitochondria, even before that, mitochondria don't move from cell to cell. They are pretty much, they belong to one nucleocytoplasmic domain.
So there will be a fixed, there will be amount of, so if biogenesis is not happening and if mitochondria is localized to a cell, then there is a fixed amount of mitochondria that is present in the cell.
Yes, absolutely.
And because, so basically, before even cellularization, there is more synthesis of mitochondria in that place?
No, we have a huge amount of mitochondria in the embryo, which is given by the mother. Okay, and so, because I have not looked at how much biogenesis there is, I can only tell you about whether there is a change in the number of mitochondria during the syncytial cycles and in cellularization. I can tell you that in the cellularization phase, when the epithelial cells are forming, the number of mitochondria do not change from at least the estimates that we have. We've published that. Yeah.
Yeah, so just the question that where are these mitochondria? Is it possible that the mother that is depositing the...
No, so to begin with, the mother deposits it, right? Then epithelial cells form. Epithelial cells have a varying distribution of how mitochondria are present. So typically, there's a little bit of mitochondria which are present throughout the cell. When there is a cell shape change occurring at this point when myosin activates in these cells, mitochondria are now enriched in this region where the cell shape change is occurring.
That's what... How?
And this is occurring because mitochondria from the base of these cells is being trafficked to apical regions of the cell. All right. So
intracellular.
it is in the cell. Yes. The transport is intracellular. It is driven by microtubules and it is occurring from the base of the cell to the apical region of the cell. All right. And I mean, this is just an example of how we can test something like this. We can test something like this because we have mutants which make really bulky mitochondria. They do not have very small mitochondria. When the mitochondria are very bulky, they cannot transport anymore on motors. They get stuck at the bottom. They do not reach the top. And then this particular apical constriction does not occur normally. So we know that it is very important. The presence of mitochondria in that location is very important for formation of this furrow. Okay, so that is essentially what I wanted to show you. That is my last slide. And just to come back to this summary from Vishas's group, that we have tried to look at how the morphogen gradients are present in this system very grossly, and what is their role in mediating a spatially regulated shape change of that of gastrulation and ventral furrow formation, where apical constriction happens. What you see is that apical constriction happens in these cells on the ventral furrow in a manner that there is constriction and stabilization of myosin, which is seen in these regions. Constriction and stabilization of really the cell area. And during those constriction and stabilization pulses of the cell area, there's aggregation of myosin, which continues to occur. And this is mediated by twist and snail. Snail is very important for initiation of the pulses. And twist is important for stabilization of the pulses. Twist is also important for transmission of the force along the entire tissue. All right. And since someone asked as well that, what is twist exactly doing? I want to leave you with the idea that there could be a lot of other regulators which remain to be discovered in how snail and twist might control these constriction, stabilization pulses of the area, the clustering of myosin which takes place in these cells. And there will be, hopefully in the future, a lot of nice studies which allow you to try and think of what is the role of other organelles in this process, or what is the role of other actin regulatory proteins, microtubules in this process, and so on and so forth. That's the end of this particular lecture.
Thank you so much. So any questions?
They have asked a lot of questions. Yeah, sure. Go ahead.
So there is gastrulation going on
Yeah.
this way, right? And also during gastrulation, the mesoderm which is present in the bottom also moves up and then gets gastrulated. So how does that exactly happen?
So maybe we can go to these images which summarize very early on the kind of shape changes which are occurring as gastrulation takes place. So in this case, for example, to begin with, the cells apically constrict, and we essentially only looked at this phase, right? And after this, there is a second shape change which takes place. You also see that the nuclei which are present here are also moving more towards the bottom. This is the region in which what we see is the accumulation of mitochondria right here. All right. And the nuclei move at the bottom. There is a further shape change which takes place. And then after that, there is a presence or redistribution of cells on the inside of the so-called ectodermal cells, right? So there are some studies, and I do not remember them, I will, outright tell you, which talk about changes in polarity which occur in these and these phases. So what this is allowing you to test is how the cell now removes its interaction with cells of this layer and goes inward, but maintains its interaction with its own cell types, right? And then later on, it completely changes shape and forms cells which are present towards the inside. They are also, so epithelial in nature, but they are now present inside. They start dividing as well. So there is a lot of knowledge which is also there in these stages about how polarity changes, but I don't have information on that right now. They are fairly recent studies.
Can you just show the previous slide? So yeah, you have mesoderm, the to-be mesodermal cells right at the bottom. But in the next slide, after gastrulation has taken place, you see it almost envelop almost the entire inside of the cells.
So basically, along this entire region, you are seeing, so the ventral furrow is present along the entire ventral region, and then it expands in this region.
Okay.
Right? So you see the mesoderm spread in this entire axis.
So that expansion is due to division, or is it more of cell just being pulled?
Both. So cells are present in this axis in the beginning. Also, these germ cells, so the embryo does a lot to secure its germ cells. The germ cells are present outside in the posterior side. This is where the nanos, protein and mRNA gradient is also present. And when we see this activity which is going on of formation of the mesoderm, there is also a point at which the germ cells are going to be taken into the embryo so that they can be secured, and they will be the ones which will give rise to the sex organs later on. So somewhere over there. So they are responsible for the formation of the sex of the animal, and they are moving inward. So some of the movements also happen exactly to position these in the correct location.
That movement is happening along the cephalic furrow? Or
No, cephalic furrow is here.
there. So what is the other?
So there are many movements which are going on, and I don't remember the names of each of these movements. But generally, there is this which is a part of the endoderm now, which is also going inside and will give rise to the formation of the sex organs at some point in development. Yeah.
I have a question about this last part about mitochondria moving. Maybe it's a bit like, I don't know, naive biologically, but like from physics perspective, mitochondria are like energy sources for the cells, and you need to have this, there is conformation, some change, like probably the cell needs, additional energy to move these cells to kind of reshape. But is it like general thing that, in general mitochondria would, whenever there is some event of
Cell shape change.
Yeah. And is it associated, or is it somehow, specific for this gastrulation?
Yeah, so we try to review this in a study of ours where we try to look at many different contexts in which there's information about mitochondrial distribution when there is a cell shape change event. And it turns out that in many such cases, for example, in a migrating cell as well, mitochondria will be present at the leading edge of the cell. If the cell does not allow the mitochondria to translocate at the leading edge, then the migration ability of the cell does get altered. So, in, for example, synapses in neurons, mitochondria are present at the synapses. And if mitochondria do not go to the synapses, then a lot of activity of the synapses is really subdued. And one of them is to do with calcium buffering. And another could be, related to metabolites which are needed in these local regions of the cell. One of them could be ATP. The reason why I'm not saying ATP confidently is because in this study, what we did was we changed the shape of mitochondria so that they remain at the base. They do not come to the apical region. And, when we change this shape, there is no effect on its ATP generating capacity. They still generate ATP just fine. But we do not see an impact on the ATP. However, we do see an impact on the ventral furrow formation. So it is some other metabolite which is now regulated by the mitochondria. And what could that be? And one of the things which is studied again and again is the change in reactive oxygen species in that particular cell type. So what we also know is that when we fuse mitochondria a lot, so this is a schematic which shows you that. But if we fuse mitochondria a lot, then their capacity to give rise to reactive oxygen species reduces. And an optimal amount of oxidation of proteins is also needed in various different developmental events. So if the oxidation reduces, then in fact myosin contractility also reduces. And it is to do with maybe the rate at which kinases and phosphatases are getting activated or inactivated to maintain a certain level of myosin activity. So that could be one mechanism. We are trying to test that. It's something which is known in literature in many different cell types which have cell shape change analysis and the role of mitochondria with it.
Thanks.
And the ventral furrow not forming, can't it be, you were telling that it might not be due to ATP, but because you told the generation
Because
of
in this particular scenario, we know that there's a one ventral furrow defect, but there is no ATP defect.
But then can't it be more about ATP localization at apical?
Yeah, but ATP is a really small molecule.
So
It can
would
diffuse
diffuse
very
fast.
easily everywhere. But yes, of course, you could still have a concentration gradient of ATP in where the mitochondria are present. But if the mitochondria are present in a wild type manner, as they are in unperturbed embryos, then there's a lot of mitochondria which are present up, down, everywhere.
Right.
So how do you change the shape of mitochondria?
So there are, mitochondria are very dynamic organelles. They keep fusing and dividing. And the fusion and fission is regulated by this class of proteins, which is the dynamin family of proteins. There are dedicated proteins for fusion and dedicated proteins for fission of mitochondria. So one of the proteins which we study a lot, I have for many years, is a protein called DRP, a dynamin-related protein. And when we remove DRP from these embryos, then what we see is that mitochondria, which are usually these small dotted-like structures, are now present in filamentous conformation and they cannot move anymore. They become very bulky for the motors to carry them. So then they get stuck in one particular place.